Okay, I just made a terrible mistake, so I'm starting over. This is the prep for lecture 15. I would like to begin with just a little bit of terminology. We're going to move now from talking about independent assortment and genes that are on different chromosomes and assorting independently to looking at genes that are not assorting independently and the fact that they are found fairly close together, relatively close together on the same chromosome, and therefore they're physically connected to each other when they're passed on to the next generation. And so I've just included this picture because when we're talking about linked genes, we're thinking about this, like the links of a chain connect the whatever's on the other side of these chains, or even just connects the links to each other. And so when we're talking about linkage, we're talking about connection. Specifically when we use the term linked genes, we are talking about genes that are found as shown here close together on the same chromosome. And they tend to be passed on together, the particular alleles, more frequently than you would expect based on independent assortment. They are not segregating independently. There is a dog barking. I'm going to keep going because I don't have time to keep re-recording. But I want to just alert you to the idea of when we talk about genes that are X-linked, when we say X-linked, we just mean on the X chromosome. And you know, the terminology we use kind of arises historically over time. It gets used and then it becomes the way we refer to things and the way the terms everybody uses. And so sometimes there are potential confusions, such as linkage versus being X-linked. And this, when a chromosome is specified, Z-linked, Y-linked, you know, X-linked, it just means on that particular chromosome. Whereas if we talk about genes being linked without any specification of a chromosome, then we're talking about this kind of situation. So, you know, part of my job is to just kind of alert you to those kinds of potential confusions so that you can, you know, get the idea straight in your head and make sure you avoid those confusions. So we're going to talk briefly about one of the earliest experiments that was done that discovered in peas, as it turned out, the same organism that Mendel used. But in this case, they found a dihybrid cross that didn't give an expected ratio for independent assortment. It didn't give a 9:3 to 3:1 ratio. And when test crosses were done, it didn't give 1:1 to 1:1. Those kinds of ratios we expect when genes are on separate chromosomes. And so there was a pair of alleles that showed up together more often than expected based on dependent assortment, and then another pair of combinations that showed up less often than expected. So you saw two of the alleles, one for each gene, passing on together more often, and two passing on together less often. And the genes they used were for flower color. In this case, it was purple flowers versus red. So this wasn't the garden pea, actually. This was, there's decorative plants that are also pea plants. And in this case, purple versus red flowers. Oh, please don't bark anymore. And pollen shape. So this is something you have to use a microscope to look at. But the pollen could be sort of long and narrow pollen grains, or it could be a more rounded pollen grain. And so they found genes that had two different alleles for that, and another gene that had two different alleles for that. And they did crosses, and they fully expected to see the same results Mendel got with his kinds of traits that he studied, the 9:3 to 3:1 ratio. So they found, they started off with homozygous strains, pure breeding for the two traits. So purple flowers and the long, narrow pollen. They crossed that to plants that were homozygous for red flowers and round pollen. And when they looked at the F1 all had the purple flowers and the long pollen. So what does this tell us? This tells us that the allele for purple is dominant over the allele for red, and the allele for long is dominant over the allele for round. So these flowers would look like this. okay? And that's what we know. We don't know anything else. And so now what we're going to do is we're going to self-fertilize those, and we're going to look for that expected 9 to 3 to 3 to 1 ratio. And then this is what they got, in fact. They got 284 purple with long and 21 purple with round pollen. And that doesn't look like a 3. We would expect long to round to be a 3 to 1 ratio. So even just looking at these two, you go, oh, this isn't quite right. This is like more than 10 times as much as this. And then 55 red flowers and 21 that have long pollen, and 55 that have round pollen. So this is like, this is by no means is this actually the 9 to 3 to 3 to 1 ratio that you would have expected. There's like way, way more of these than you would expect. There's actually relatively more of these than you'd expect. Red and round are the two recessive traits, so we would expect that to be the 1, and it's actually more than these guys. And we'd expect these to be the 2, 3 categories, and this one to be the 9. So the fact that this is in higher proportion is consistent with what we'd expect, but the rest of the numbers are way off. And you will find, if you do a calculation to figure out what the number should be, is that this number and this number are way higher, or somewhat higher than they should be, and these two numbers are considerably lower than they should be. And we will come back to this when we talk, well, not to this exactly, but I might have some questions in the class that ask you about the categories and what you might conclude from them. And now they're doing construction upstairs. This is very difficult. I'm going to continue, and then if it turns out that the noise is too much of an interference, I'll have to redo it maybe on the weekend. So basically, Thomas Morgan, who I talked to when I talked to you about genes on the X chromosome, because that first mutation he found in fruit flies was the white gene that was on the X chromosome. And he found similar kinds of things. He isolated a strain of fruit flies that was homozygous for purple eyes and normal wings, the wild type, and another strain that have these funny little stumpy wings called vestigial and had wild type eyes. And so this is the way, just take note of how we write out the genotypes. The ones that are just the letters, purple here, are the mutant ones. And when there's a little plus here, this means wild type. The wild type is the normal one. And when we say normal, we mean like if you go out into the wilderness and collect a bunch of fruit flies, they're all going to have these normal wings, right? They're all going to have red eyes, right? It's the most common when the gene is functioning, okay? So these are the genotypes that he was dealing with. And they did this cross. They took flies that were homozygous for mutant for the 2 traits, crossed to wild type, and just remember this dot. I'm writing the cross this way because we don't know the linkage arrangement. We don't know if the genes are on the same chromosome or on different chromosomes. That's why the cross is being done, is to find that out. And so when we write it before we get the results, we write it this way. We will change the notation depending on whether we find out that the genes are sorting independently or that they appear to be linked. And so the progeny are dihybrid. They're heterozygous for both genes, and they show the wild type phenotype. So that tells us that the wild type alleles are the dominant ones, and the mutant alleles are recessive. And then we do a test cross, and this is way better than the hybrid cross done on the previous experiment, because we take those females and we cross them to males that are homozygous mutant for both of the genes. This means that we'll expect to see a 1-to-1 to 1-to-1 ratio in the progeny. And when we look at the results, I'll talk a little bit more about why this is really helpful and what you can do from it, because we're going to be talking about actually mapping the genes in a few minutes. Probably not in this tape. But so the test cross is really useful because the tester parents are not contributing anything except recessive alleles. So if crossing over is occurring or independent assortment in that parent, you don't detect it because even if there's crossing over between the A and the B gene or the purple and the vestigial gene, the genotype that is passed on is exactly the same. It's recessive. So the only thing you have to think about is there crossing over, independent assortment, etc. going on in the parent that's heterozygous? And then we just look for recombination or independent assortment or crossing over in that parent. And we don't have to think about it in the tester. It's more complicated arithmetic that has to be done if there's crossing over and stuff occurring in both parents, right? Okay, so these are the data that Morgan's, probably not him, but his one of his grad students would have generated this. So wild type for both traits, 1,339. Mutant for both, almost 1,200. But then wild type for the eye color, but mutant for the wings, 161, way less, right? Way fewer. And then mutant for the purple, but wild type for the wings, 164. So we're seeing one category, one pair of categories that are more frequent than expected, and one category that are less frequent. And they're paired in a very important way. So this category has the two dominant alleles, and this one has the two recessive alleles. We call these reciprocal classes because we have a dominant and a recessive allele for each of these genes coming from that heterozygous parent. And if you pass on the two dominants in one gamete, the other gamete should have the two recessives. So we wouldn't expect purple wild type and vestigial wild type to be 1,339, and then purple plus wild type and vestigial mutant to be that, because then that would just suggest that purple wild type is being passed on more frequently than normal. It has to be the reciprocal classes in order for this to be evidence for linkage. And I won't go on about that too much, but it's very important when you're, to pay attention to that. And we'll talk about it again when we talk about trihybrid crosses, which we won't be covering until the next lecture. And then the other two categories, you have a dominant allele for purple and a mutant for vestigial, and then there's the wild type for vestigial and the mutant for purple in the other category, and these are paired. So whatever you're missing in this category, you have in this genotype, you have in this one, and vice versa. Okay, so it's very important. Sometimes the numbers are closer than this. It's pretty obvious which categories should be grouped together to discuss in this case, but if the numbers were closer, you would want to pay attention to that. And also, as I said, I'll talk more about that. It's very important, especially when we're looking at trihybrid crosses. So what do we see from this? We see that there's a pair of combinations of alleles that are much more frequent and a couple of that are much less frequent. Now, it turns out that if you look at the frequency, the ones that are most frequent, they are similar to the original parents, that P generation, because you had one parent that was mutant for both and one parent that was wild type for both. And that's important. These are seen more often than the other classes, and that allows us to speculate on an interpretation, right? So this is how we would explain it. We would say, okay, so this parent, and I've color-coded it, hopefully that's helpful if you can see color. This parent had the two recessives, and this parent had the two wild types. Here's our dihybrid, here's our test cross, and I'm just looking at the alleles here. This is the phenotypes, right? And it's only showing what came from the heterozygous parent because the tester parent is only giving us recessives. So we see these two from this parent, and I've color-coded them that way, and these two from this parent, and then these are a mixture of non-parental types, or what we would call recombinant types. And this would suggest to us that the alleles are actually physically connected to each other on the same chromosome. so that when this parent makes gametes, most of the gametes are going to have either the two dominants because they're connected to each other. Whoops, no G there. Or these ones. And then the question would be, well, okay, but there are some that are the other combination. How does that happen? And that happens from a process that we call crossing over, which I'm sure most of you have heard about already. But it is a case where there has been a breakage and a swapping between the chromosomes. And it doesn't happen very frequently, but where the purple mutant allele and the vestigial wild-type allele are now connected together because these two parts of the chromosome have been swapped, and that these are the less frequent ones. Okay, hopefully that makes sense. I'm just going to look at the next slide. Yeah, so let's just rewrite those genotypes. And this is using the correct notation that you would use for genes that are actually linked. And because we know what the parents originally were, we know that this is a cis dihybrid, the two wild types on the same chromosome, which I'll talk about in a minute. So we know the exact arrangement. And then when we write it out this way, I think it's easier to see that these non-parental recombinants have come from a crossing over event. And you can even, which we will do, boy, it's noisy today. What we will do is calculate the percentage of recombinants, because we can say, okay, it looks like there was almost 11% recombinant from this category. When we expected, if the genes are on different chromosomes, we would expect there to be equal numbers, 50% recombinants. And I will stop there, but I want you to consider, suppose we do this cross differently. Suppose one of the parents is wild type for one gene and mutant for the other, and then the other parent is the other way around. Think about and predict what the outcome should look like, and when you view the next recording, we'll start with that.